Welcome everyone to our webinar, and this is brought to you as part of the Mental Health Research Consortium Neurotherapeutics summer activities. And we're really thrilled to have you here as we're going to talk about a crucial topic that has gained significant interest in academia. So the landscape of drug discovery and drug development, with the academic sector has been rapidly evolving, and consequently there is an urgent need for academic lab to learn and adopt new technology and instruments that are capable of performing tasks with higher speed and efficiency. And additionally, there is also an important aspect to it, which is the integration of miniaturization of these equipment that has proven really invaluable and could in the future enhance production and cost and lead to cost reduction. So today we are privileged to have two distinguished experts who will discuss the potential of their product and shed light on their utilization within academic labs. And leading the way is Dr. Alessandro Porro from Element SRL, who will be our first presenter, followed by Team Strassmaier from Nanion. And without further delay, Alessandro, you can start.
Yes, so let me share the screen. Okay, you should see the presentation now, right? Okay, great. So first of all, Fernanda, let me thank you for inviting us to this seminar. And so as Fernanda has anticipated, the main protagonist of the talk is ePatch, which is our patch clamp amplifier, which embeds this technology that allowed us to miniaturize the device and make it as small as possible, so even less than a smartphone. The outline of the presentation, yes, I will do the talk in three parts, let's say. So first few slides is a brief presentation of the company with the core technology and the products overview. Then, of course, the focus on the ePatch amplifier. And then, since the topic of this seminar is neurotherapeutic development, I've decided to include a few slides to talk about this project in which I'm involved, and that is running at the University of Milan, where I also work, and that is focused on HCA1 channel variants linked to human epilepsy. So let me start with the first part of the presentation of the company in a few words. So the company was established in December 2013, so almost 10 years ago. We are currently 17 employees, and we have customer worldwide. We have local distributors in the US, of course, but also in China, Korea, Japan, Mexico, and Central America. We are based in Italy, in a small city called Cesena, which is in the so-called famous Motorland, so that you might know, where the Ferrari supercars and Ducati motorbikes are designed and produced. So the core technology of our devices is based on microchips. So CMOS microchips that enable low noise current readouts. This allows us to offer a wide range of devices for several applications, to ranging from patch clamp to single channel recording in lipid artificial bilayer membranes, but also for solid state nanopores, and also custom developments in partnership with other companies to design and develop products for, again, lipid bilayer membrane application and other. So the simplified scheme of our product, that is in some way shared in all our products, is represented here in this scheme. So in the small box of our amplifiers, there is the microchip, which embeds, of course, the low noise amplifier and the ADC and DAC converter. And then all the data are processed by an FPGA, which is managing the data coming from the microchip. And all our products are powered by a USB cable, which is directly connected to a computer that can even be a laptop. So the only power source that is needed is just the 5 volt of the USB. So this is a slide showing the products that we offer these days. So the very first product that was designed and developed by Elements, which is this E1 that you see here on the top left, and it was designed for single channel recording artificial lipid membranes, and that we sell also as a kit with all the components that you need to form the artificial lipid membranes, where you reconstitute the ion channel that you are interested in. And then from an evolution of this first E1, it came out the ePatch, where we have integrated, of course, all the circuitry needed for the compensation that are not needed when you record single channel activities. So I'm talking about the CFAS, C-slow, and R-series compensation, for example. Then we have also products as this nanopore reader, for example, for recording solid state nanopores, but also biological nanopores. We have multi-channel amplifiers. And the last board that is this 10 megahertz amplifier. So a very high-speed amplifier designed to record very fast events in the range of a few microseconds. That, to our knowledge, is the only 10 megahertz amplifier which is available on the market now. So, since it might be of your interest, I would like also to spend a couple of words about these 2 devices that we offer for single channel recording in planar lipid bilayers. So in both cases, so in the case of the BLM kit and the nanopore reader, we also provide a recording chamber, which consists of 2 semi-compartments separated by a central septum, where is drilled a small hole, which is in the range of a few hundred micrometers, that is physically the place where you paint the artificial lipid bilayer membrane. So by adding then your ion channel or membrane protein that you want to study, you are able to record this nice single channel activity. And what I would like to point out from this slide is also the very good signal to noise ratio that you can get from these devices, as for example, this membrane, artificial membrane here at 5 kilohertz is producing only a few hundred of femtoamps of current in noise. So as I said, also we have also electronics which is embedded in other products. For example, we have a partnership with Nanion, and we provide them the amplifier for some of their products dedicated to the single channel recording in artificial lipid membranes. So I'm talking about the Orbit Mini, for example. And so let me go now to the protagonist of the talk, which is the ePatch amplifier. I always start with this slide here, which shows how it is usually made, a traditional rig, patch clamp rig, where you have usually these big and bulky boxes, which are the amplifier and digidata. These are usually quite expensive, and they are separated. So you have one box for the digidata and one box for the amplifier. Then you also, of course, you need an acquisition software that many times is sold separately, and usually is quite expensive too. And so this leads to have an expensive rig at the end with a need of a lot of space, because these devices are quite bulky. And also this is what is hard for, especially for the beginners is the installation, because as you probably know better than me, this requires also the connection of many cables and the configuration between the DD data, the amplifier, and then together with the software. So what we have made from our side was to miniaturize everything. So thanks to our microchip, now everything is embedded in this small box that, as I said, is even smaller than a smartphone. And the technology of the microchip allow us to keep the price quite low. So a reduction of price thanks to the large scale production of microchip, compactness, and simple installation, which are the pros of our device. Because once the amplifier is installed directly into the micro manipulator of the patch clamp rig, you just need to connect the USB cable to the laptop and a few other cables in there for the grounding and the reference electrode, and you're ready to patch clamp the cells. And of course, last but not least, is the fact that the acquisition software that is dedicated to this device is included in the price. So in this slide here, I would like to quickly focus on the main features, just to say that the device is capable of recordings in both voltage clamp and current clamp mode. Of course, it is equipped with all the compensation that are needed for both the modalities. So C-fast, C-slow, and R-series compensation for the voltage clamp, and the pipette neutralization and bridge balance compensation for the current clamp. You have the possibility to collect samples up to 200 kilosample per second, so more than enough to detect the fastest biological events, and an available bandwidth that is up to 100 kilohertz. You have 4 current ranges or gain available that allow you to record currents up to 200 nanoamps. And for the current clamp mode, we have also introduced this modality, which is present also in other amplifiers from competitor, which is the true zero current mode. That is, when enabled, it physically detach the circuitry needed for the stimulus, and this allows you to record the real and true membrane potential of the cell that you are measuring. Last but not least is the fact that it saves file as an ABF format. So maybe some of you are also already familiar with this file format. So you can still use your software that they are maybe using now for data analysis. So talking about the software, we have designed and developed, and we keep on developing this software interface that is needed to control the amplifier that we have called ePatch. And the design was based on user feedback. So based on the feedback of the scientists, we have designed and developed the interface. From the website, if you might be interested, there is also the possibility to download a demo version of the software that you can run even without the device attached to the computer, so you can play and explore the features of the software. Main and interesting characteristics of the software interface is that there is a soft switch between the voltage clamp and current clamp modes. So switching from the one modality to the other does not affect the cell health, and this is important in some experiments. The compensation settings are both either automatic or manual, in the sense that you can automatically compensate for the C-fast and the C-slow capacitance, but you are still allowed to manually adjust the compensation setting if the result of the automatic compensation is not good for you. The acquisition manager embeds the protocols that some of the protocols that we offer are pre-designed, let's say. So this helps maybe the beginner users. So, for example, we have included this protocol that we have called seal test that is used usually by the user to check the passive properties of the cell. But of course, there is the freedom to create your own protocol by using the waveform that are available. So we are talking about voltage steps, holding voltage, ramps, sine waves, and whatever you can imagine to draw and design your waveform stimulus. Of course, the passive properties of the cell are always displayed in real time, so that you can also check if the cell is the same at the beginning of the protocol. And we have also introduced real-time analysis tools, which are sometimes useful to decide the direction of the experiment. So, for example, you can while you're collecting data, you can build IV graph in real time, but also histogram, fast Fourier transform, and you can also keep track of the noise during the recording. This, as I said, is the protocol editor. So this is just to show that you can design your preferred waveform without basically any limitations. So you can add whatever step you need in your waveform. You can add ramps, sine waves. You can change the amplitude of the steps and also the duration step by step. So duration and amplitude can change over time. So this is a sped up video, which is showing how easy it is to collect data during an experiment. And this is, for example, the recording of an X cells which was transfected with the potassium channel, voltage gated potassium channel. As you can see in a few clicks, I'm running through the patch clamp configuration. So from cell attached to whole cell by compensating the C-fast and C-slow capacitive components. And at the end, you will see the HCN channel current running on the screen. And at the same time, the building of an IV graph from the fitting of the dot of the points of the data of this graph, it is possible to get very fast and very quick the reversal potential of this current that in this condition fits with the solution that were used for the experiment. And as you can see here, for example, the reversal potential is then plotted into the is reported in the lab book, which can be exported then as a TXT file at the end of the experiment. Last is the presence of a port on the back of the amplifier, which is a digital out port, where you can connect this BNC cable and connect external devices, so for example, perfusion system or lamps. And this works with the TTL signal, of course. And good thing is that you can trigger the TTL signal with the protocol by using cursors that you place within the waveform that you use as a stimulus. And basically by, oops, these two videos are supposed to start at the same time, but this is just to say that you can trigger, for example, this lamp at a specific time of the waveform of the protocol. So this is, for example, used in optogenetic studies. So the last slide regarding the software is to say that the software is under a continuous development, and we are also always open to listen to the requests or the needs of the users, of the scientists, to develop new tools and new features in the interface. And also, you can ask us to provide a DLL so that you can also develop your own acquisition interface, your own acquisition software to control ePatch amplifier. So important test that we did was to perform to validate ePatch amplifier from a scientific point of view. So first thing that we did was to collect one of the most one of the hardest current that you can measure in cells, which are sodium currents. They are very fast, and a run compensation in the capacitance in the series resistance compensation may affect the recording and may lead to wrong measurements. So we have recorded some traces, and we have compared them with other amplifiers that are available on the market, and we found that the data are totally comparable. We did the same thanks to this group. They are expert in recording the firing activity of neurons. We have also tested the capabilities of the current clamp mode, and they state that the recordings that are collected with the ePatch are totally comparable with those collected by other amplifiers. So another nice test that we did was to investigate a possible interference given by other instrumentation within the rig. So, for example, with the perfusion system. So what we did was to record this the current of P2X2 receptor expressed in X cells, and as you can see here, the switching on and off of the pinch valve of the perfusion system, which is used to deliver the ATP to the receptor, was not affecting the current of the receptor, which remained very clean during the whole recording. Then, of course, we have did also some tests in inside out, and this is just to show that we do not invert the sign of the current in inside out recordings, but we are thinking to introduce a button to help maybe the beginners of the technique that are not familiar with the fact that in inside out recording the sign of the current is reversed. So we will soon introduce this option in the software interface. Then another test here, as shown, is we have measured the dose response curve of this potassium channel, HCN channel, which is opened by which opening is triggered by the voltage, but is still regulated by the binding of the cyclic MP, which is affecting this activation curve, shifting the curve towards more positive potential. And we have built this dose response curve of the cyclic MP for the channel, and the results that we obtained in the EC50 is comparable to those that are published in the literature. Of course, last was to test if the signal to noise ratio was good enough to record single channel activity, and the answer is yes. So we have overexpressed this small viral potassium channel in X cells, and we have made some cell attached patch from which we have measured this nice single channel activity, and the single channel conduction that we got was again comparable to those reported in literature. So in the website, if you are interested, there is a list of some publication that include ePatch as an instrumentation in a material method. We are pretty sure that there are more, but those that are in the list are those that we know to exist. And just a focus on a few of them. So in this case, for example, this is a nice paper. I'm a co-author of this paper, so I'm pretty proud of that. Published on Molecular Cell some month ago. And here the structure of HCM4 channel was resolved for the first time, the cryo-EM structure of the full protein. And by using ePatch, we could also highlight a new pathway for the cyclic API transmission from the cytoplasmic side to the transmembrane region, which involves the presence of this coordination site for a magnesium ion. Another nice publication is this from eLife in 2022, where lamotrigine drug was tested on sodium channels. And also this other paper from this group who has used ePatch to record single channel of this C-ring protein from proteoliposomes. And the protein was added as a purified protein in the patch clamp pipette. So thanks to its low cost, ePatch is also suitable for teaching activities. And what I do here at the university, we organize also these practical activities for undergraduate students, mainly they are master students. So we have built 6 units, patch clamp rigs, in this lab, which is the basement of the university. So it is a good location because in this way we can keep the vibration coming from the environment quite low, so we don't need the real anti-vibration table that would make the cost of the rig too expensive for the university. So usually there are 10 or 12 students divided in a group of 2 or 3. And as you can see, the rig is very easy. It's a small microscope and one of these old still mechanic manipulator, and ePatch, of course, with the laptop, and this metal base used to dampen the vibration coming from the environment. And what we do here is we teach to the students the ion channel biophysics. So, for example, we use this neuroblastoma cell line that we have in lab, which is nicely expressing this sodium current. So we teach the student to record this current and to analyze data to get the activation curve, the inactivation curve, for example. And also the past year, we have done successfully the recording of action potential firing from neurons extracted from neonatal rats. And this is was very funny for the students, and also for them to really record a trace and see the spikes running on the screen. They really understand how an action potential is made, instead of doing only by reading a book, which is always, I think, kind of limiting. So, conclusion of this second part, let's say, that we believe that ePatch provides and enable reliable measurement and high quality data that are comparable to those collected with other patch clamp amplifier that are on the market now. And again, thanks to our technology, so to our microchip, CMOS microchip, we can keep the cost very low, and the installation of the setup is very easy. So we can, we are happy to say that with the ePatch, the patch clamp experiments are, we think, accessible to everyone, or at least to more people than before. The last slide. So let me thank you for your time and your attention, and I'm happy to answer any question from your side.
Thank you so much, Alessandro. And yes, we have maybe, I would say, 5 minutes or so, 5-10 minutes for questions. If anybody has any questions for Alessandro, it's time now. It's the time to ask. I have a couple of questions, but I'm going to wait just in case anybody has anything else.
Yeah, and of course, Fernanda, if some question will arise in the next days, so you can give my contacts to this person, and I will be happy to chat with him or her.
Great. So, okay, so I have two questions actually. One is, I guess it's in the opposite order. One is about a technical question. So you set up all of these ePatch rigs at the basement essentially. And you said that you are not using any anti-vibration table. I see that you do have a pretty thick metal base to maybe mitigate some of maybe residual vibrations that they might exist. So what is your experience with that? Obviously, that if there is a way to even, to cut even further down the cost by, avoiding the anti-vibration table. I know your products are like super competitive, money-wise. But if we could find a way to really get rid of the anti-vibration table, that would even. So what is your experience here? So is this like, what type of what basement? Is it underground or is it's
yes,
underground? Okay.
Yes, it is. So, Fernanda, the answer to your question is pretty hard because this really depends on the environment. This must be tested. Of course, being at the basement is an advantage because, at the 8th floor, vibration are much bigger than at the first. And so if you have the possibility to set up everything as down as you can go in the building is better. But in this case, for example, this metal base is working pretty well. So of course, once you have the cell under the cell patched, you don't have to touch the table, you don't have to jump, you don't have to run in the room, but students are not supposed to run in the room during the practical course, so should be fine. Must be tested. What I can suggest you is that by using tennis balls, and believe me that it works, that you can place just under the metal base, so between the table and the metal base, can help to further reduce the vibration coming from the environment.
So, as you know, we purchased one of your devices, and we're still waiting for an anti-vibration table. Because in our case, there is really not a possibility. We are on the 7th floor nominally, but it's probably higher because we have mezzanines in between. And in Texas, as far as I know, there's really nothing truly underground. I know the reason I ask, because I know typically in universities in Italy, you do have basement. There are true basement and basements. So that's why I ask whether it is. So, but that definitely, as you pointed out, for teaching purposes, that's tremendous. It's a tremendous advantage. In fact, there is a major need in the US for electrophysiologists. Even if Nanion is producing robots, there still needs a major. There is still a major need for electrophysiologists, and being able to possibly opening up teaching opportunities to undergraduate, which will correspond to like university students in Italy. That would be terrific. So anyway, so that was a question I had. The other question you mentioned initially, a new amplifier, excuse me, the 10 megahertz amplifier.
Oh, yes.
Yeah, you said that is the fast that can detect super fast event, and you say that is in the fastest on the market. So what type of use do you have for this amplifier?
Yes, so this is mainly designed for solid-state nanopores applications, because in that field, in some cases, a very speed collection of the signal is needed. For example, as you probably know, the sequencing of the DNA through a nanopore is something that is running in these years, in these days. And in that case, you need to detect very fast events, which are in the range of microseconds. And the traditional amplifiers that are available on the market now, they can go up to some hundred, in the best cases, of kilohertz, while for very fast events, a much higher bandwidth is needed. So this is the reason why we have created this 10 megahertz amplifier, which in the community of the nanopore scientists and application is quite desired, let's say. So they're pretty happy to have this new tool for their for the detection of their signals. For the biological signals, maybe it is less useful, because to go up to 10 megahertz bandwidth, of course, the noise will increase. So probably in the future there will be also the possibility to better manage this signal in order to reduce the noise and to study the ion channel kinetics at the single channel level, for example, in a way that was never done before. But this is
that is exactly that was my question. Yes, I think that's that is the dream. Yes. There still is a gap, a knowledge gap. And yes. If we could potentially measure directly these kinetics that so far have been only be modeled, with differential equations or model different model, that would be fantastic to experiment and measure these things.
That would be great, but still, I mean, if you want to do that, you have to do at the single channel level, and so far the noise at that bandwidth is still too high to detect the single channel opening.
Okay, this is great. Any questions from the audience? I do see in the chat from Parsa, Armen, very good presentation to Alessandro. Any other comment, any other question?
I have a very brief question.
Thank you, Elena. Yes.
But first, I would like to send my best regards to Anna Maroni. I didn't know that you work with her, and I know her very well, so please send her my regards.
Thank you.
And my question is like that. So your ePatch amplifier looks really fantastic. Even I would say too good to be true. So my question is, what are its disadvantages compared to more conventional things on the market like ePatch or High amplifiers? Are there any disadvantages?
So, I mean, yes, there are. And so one, for example, is that I think that is the main limitation. Now, I don't know why my presentation is blocked, but the point is that I told you that on the back of the amplifier there is a port which is dedicated to the digital out signaling, so a TTL signal, and so far only one port is available. So this means that you can control only one device per time. So if you need to control, for example, a lamp and at the same time a perfusion system and to trigger everything with the stimulus protocol, you cannot do that because you have only one exit available. And another small disadvantage is that the signal to noise ratio is a little bit worse compared to other amplifiers. So if you need to record the very small, very small currents, so maybe ion channels which are in they have a single channel conductance, I don't know, of a few picosiemens. In that case, it might be hard to record those activity with our with our amplifier, with our ePatch amplifier. But for the rest, as far as I know, everything can be made with our amplifier.
Thank you. Thank you.
You're
Looks
welcome.
good. Looks great.
Can you please tell me your name so that I can, Elena? Okay, sorry.
Yeah, I can put it in the chat. Yeah, where we are.
Yeah, I can also send a common email.
Okay, so that I can say the right hello to Anna.
Yes. Okay, I mean, this is great. Thank you so much. And I think now it's Dr. Strassmaier from Nanion, Team Strassmaier time. And yeah, we'll go with Nanion. Thank you. Thank you so much, Alessandro, again.
Thank you.
Okay. All right. You're able to see my presentation, I assume. Is that correct?
Yes,
Okay.
and I can see and hear everything.
Okay, perfect. Yeah, thank you so much for the invitation. It's very nice to be able to talk to everybody a little bit about Nanion technologies. And what I'm going to focus on today is the instrument that is the one that I work on for the most part here at Nanion Technologies and the applications side of things. So just to give you a little bit of background, this is a great time to be talking about Nanion because we just had our big 20th birthday party bash. It was company was founded back in 2002 based on work that was done. Let me see if I can get are you able to see my mouse at all when I mouse over? Okay, perfect. Yeah. So Niels Fertig, who is our current CEO of the company, his research, PhD research, eventually led to the technology that led to the first product that was launched by Nanion back in 2003. So the first product we ever launched is called the PortaPatch, and that was back in 2003. So 20 years of the PortaPatch, and it's still going strong. And so all of the core of this technology that Niels developed, along with help from Andrea Brueggemann and Michael George, who were the CSO and chief technology officers of the company today, is the idea of running patch clamp without a patch pipette. And so instead of a patch pipette, what you have is a planar piece of borosilicate glass. And what he developed is technology to create micron-sized apertures in this planar piece of borosilicate glass. And then that acts as the patch substrate. And so in 2003, we had what was billed as the smallest patch clamp rig in the world. It really has a very small footprint. But it also opens up the possibility for going to higher and higher throughput. And so in 2006, they launched an 8-channel version of an automated patch clamp system. And in 2013, the very first 384-channel platform was launched. And there's been subsequent improvements across the whole line of patch clamp. But just wanted to also point out that Nanion Technologies, while it's core technology since its founding and continuing until today has been automated patch clamp. We've also launched other product classes such as what we call the Surfer instruments, which is solid supported membrane technology that allows you to automate the recording of signals through electrogenic transporters. We also have, as Alessandro alluded to, a range of products for recording single channels or doing work with artificial bilayers. And this is the Orbit Mini, which would record from four partitions at a time, and the Orbit 16 with the temperature control, which can do up to 16 at a time, and our vesicle prep for assisting with, creation of GUVs, etc. And lastly, we have our cell monitoring line, which is largely based on impedance measurements. And the original instrument in this line is the Cardio Excite, which, as the name suggests, it was for measuring cardiac tissue. And then the add-on, which is the Flex Site, which allows for enhanced maturation of cardiomyocytes, as well as actual direct force measurements, which is pretty cool. And then coming soon will be the launch of what we call the Atlas, which is another more broad-based cell monitoring system with 696 well plate configuration. And what it's doing is taking advantage of the ability to look at impedance signals across a very wide spectrum. And so you can get signals from everything from G protein coupled receptor signaling to, a barrier formation and things like that. But just so you know how the automated patch clamp fits in to what Nanion Technologies offers. Now, going back to the topic of today's seminar, which is miniaturization, really the miniaturization that is achieved by going to the automated patch clamp is based on the idea that instead of having pipette-based electrophysiology, which is the classic manual patch where you need a skilled operator, a vibration isolation table, a microscope, where you bring the patch pipette down to the cell. What we're doing in planar patch is dropping the cells into the solution and using suction to draw the cells to the aperture. And so, and Alessandro had an awesome photo of a patch clamp rig. Here's a cartoon version of that. So you have to have a rack of instrumentations with your A to D converters, your amplifiers, your oscilloscope, the vibration isolation table with its Faraday cage, and a skilled operator to do all these measurements. In contrast, with the PortaPatch, would fit on your desktop in between your computer and your monitor. The other important thing about going to the planar patch configuration is that in addition to making everything smaller, more compact and easier to use, is that you can create an array and get much higher throughput. That's why I say automated patch clamp yields miniaturization and throughput. So the SyncroPatch 384 is our highest throughput automated patch clamp instrument. It works based on a consumable chip that has the exact same footprint as a 384 well plate that everybody's familiar with from molecular biology. The thing that makes it special is the floor of each well is a piece of very thin planar borosilicate glass, and it has a micron-sized aperture in each well. Now, we actually have versions of these consumables that have either 1, 4, or 8 holes per well. So you can actually record from an ensemble of cells of up to 8 cells at a time. And I could also mention that we have different hole sizes. So you have small and large hole sizes based on what kind of cells you're patching. If you're T cells, you would want a smaller hole, for example. So SyncroPatch also includes 384 individual patch clamp amplifiers. They come with both voltage and current clamp functionality. The chip itself becomes housed inside what we call the patch clamp module that has connections to all of the amplifiers. And this in itself is integrated into a commercially available Biomech i5 I-Series liquid handler. So Nanion's really good at making patch clamp technology. We're not going to reinvent the wheel of liquid handlers. So we do an integration into this system that works quite nicely. And the same thing is true of the patch liners. We'll touch on the patch liner just briefly because the patch liner was used in parallel during all of these experiments that I'm going to describe later on the neurons. It's the same patch substrate as your traditional patch clamp. So it's borosilicate glass. And the other interesting thing about planar patch is it facilitates internal perfusion, which, as anybody who's ever done manual patch knows, that would be very difficult to achieve a complete perfusion of your internal pipette solution. So that is something that's really powerful. And by combining multiple additions of compounds to each well or subsequent additions, you can achieve very, very high throughput in data collection, up to 20,000 data points per day. So one thing that the SyncroPatch does is it provides you with this high throughput measurements. And so that naturally lends itself to target-based screening. So if you want to develop drugs for a particular target, I'm just going to use this example as an NMDA receptor assay. That's something that the SyncroPatch excels at. And so what's really important here is that we're utilizing engineered cell lines. And in this case, a cell line that expresses the NR2B subtype of the NMDA receptor and from Charles River. And that's really important because the cells that you use, the quality of those cells, will provide you with a quality assay. Whereas if your cells are not healthy, you're going to run into trouble. And Charles River, I just wanted to give them a shout out because they do produce really, really nice cell lines. And there's a number of other companies that do so as well. If anybody has questions about, favorite cell lines, give me a call. So in this case, what I'm doing is I'm applying a quality control cutoff, saying I want to have at least 500 mega ohms of seal resistance on a per cell basis. In this case, I'm actually running a chip that has 8 patch holes per well. And the average seal resistance that we're getting out of these cells is 12 giga ohms on a per cell average. And greater than 98% of these cells pass that threshold. Now, this is a really good assay, but in general, most of our assays on engineered cell lines come out at 90% success rate is pretty routine. The other thing about the SyncroPatch is that it can coordinate the application. And so this is a ligand-gated channel. It's an excitatory receptor that opens in response to the binding of glutamate. So what we're doing here is coordinating the recording, the electrical recording of current with the application of the ligand. And another thing you probably know about NMDA receptors is that they're affected by magnesium in the external solution. So magnesium acts as a pore blocker. And so when you are holding the membrane potential at, like, let's say minus 80 millivolts or minus 90, you will not see any current through the receptor, even when you apply glutamate, because of blockage by magnesium. So what we do is a little trick here is when we just prior to application of the glutamate is we step the membrane to plus 40. That removes the magnesium block and allows you to see this increase in current that happens when you pipette on glutamate. And then we have what we call a stacked addition, where this first addition of 5 microliters of glutamate is followed by 5 microliters of washout. And we have other little pipetting tricks that I won't go into, which allows us to get reproducible application of glutamate over several additions. And what's shown here in this next panel over is the overlay of 6 additions to the same concentration of glutamate over and over and over again, just showing that you can get really very reproducible signals over time, which is very important for screening purposes. So if you want to screen for substances that act as a blocker or substances that act as a potentiator of that current, ideally what you want to do is establish a baseline of your signal in the absence of any modulators, and then come in with a screening set of modulators, pre-incubate them with the cells, and then add glutamate additional times in the presence of your test modulator. And that's what's shown here in this panel down here. So I have 3 applications of glutamate, which are giving me 2 nanoamps or so of current. And then I have this incubation phase in the middle. Then I come in again. In this case, it's just glutamate again with DMSO as the vehicle. So we're seeing that the current or the signal remains stable over time. What I'm not showing here, what I'm showing on this panel here with all the wells that are displayed, is that I'm putting on glutamate at different concentrations. But what I'm not saying is that actually for each concentration of glutamate, I'm doing 2 experiments. One in which I'm applying vehicle controls in 2 columns worth of wells, and one in which I'm applying a high concentration of an antagonist, so I can get boundary points for my assay. So I can have the vehicle control and the full block, and that sort of acts as the window for my assay. And that allows me to do some important calculations about how well the assay is performing. And part of that is just based on purely on how much of a change in signal do you see in the case of vehicle control. So in the vehicle controls at 1, 3, 10, or 30 micromolar, I'm comparing the 3rd application of glutamate before application of vehicle, and then the 2 applications of glutamate after vehicle. And in all cases, there's very little change in the current amplitude. And that's shown here in the table, where you see the vehicle effect in terms of block. Let's say there's a 13%, for 1 micromolar, I see about a 13% reduction in the current amplitude. And so that's pretty minimal in general. In contrast, what you see in the case of MK801 is we see the nice current amplitudes. And then afterwards we see, and it's a little bit hard to see, but this light blue trace down here is showing the complete block of the signal. And the effect of this MK801 antagonist is in the range of 97-98% block. There's an important metric that people use for developing screening assays, and that's called the Z prime. And the Z prime is basically what it's doing is it's comparing the mean and the standard deviation of your two boundary conditions, the mean and standard deviation of your vehicle control. So if you look at this plot down here, we see there's some scatter in the vehicle wells, and there's very, very minimal scatter in the full block state. But what you're really doing is comparing the mean, the distance between those two means, and you're factoring in the standard deviation in both cases. Now, if you have a perfect assay, which was absolutely no chain, no variation at all, you would have a Z prime value of 1, and that's always impossible. But in general, for a screening assay, if you're above 0.5, that means you're going to reliably avoid getting false positives at, let's say, a 50% reduction cutoff. And you can really see that from these data here. So this is just a way of saying that, the SyncroPatch is really good at doing high throughput patch. And that's been established for a long time now. And it's not uncommon, to be able to develop this type of assay for a wide range of different kinds of channels, voltage gated channels, as shown here, ligand gated channels, and there's a variety of other ion channels, trip channels that are also have been shown to be amenable. And it's just the list keeps getting longer and longer, but this is all with engineered cell lines. People are really interested in pushing the boundaries of the technology further. But before I get to the beyond engineered cell lines, I just wanted to do a real quick mention of other uses that are really valuable in terms of the traditional engineered cell line work that's really works extremely well in the SyncroPatch. So I mentioned the target-based screening, but we also have cardiac safety testing. That's really an important avenue of use for the SyncroPatch. Basic research, and I forgot to mention, we also have optogenetic add-on for the SyncroPatch, so you can actually do light stimulation for light-gated ion channels. But one other really interesting one is ion channel variant testing. So with the advent of high throughput sequencing, there's just a vast trove of ion channel variants that are shown to exist in the human population. And people are using SyncroPatch to integrate this vast array of genetic information to, unmask what the actual functional significance is for a lot of different ion channels, and to predict whether they should be, harmful or pathogenic or neutral or whatever. And then on top of that, those variants then can be screened with the lists of known drugs. And, in some cases, you will find that the information you get out of the SyncroPatch can guide, decisions on what drugs might actually be useful for certain epilepsies, et cetera. But beyond the engineered cell lines, there are the new frontiers. And a big part of that is iPSC-derived cells. So we've run a number of different types, cardiomyocytes and neurons for sure, and microglia. But there are more and more iPSC-derived cell types. I'm going to talk today in the second part of the talk about neurons. But we've also have a lot of interest in patching primary cells. And we've had really good success, a collaboration between a Nanion headquarters and a group at the University of Göttingen, where they've actually been quite successful in patching primary cardiomyocytes from pig. We've also had a group that's done a similar thing with cortical neurons from rat. And then another thing, which is more recently becoming of interest for the SyncroPatch, is organellar patch. And this is a paper down here, which is actually lysosome recordings done on the PortaPatch. But since the core of our technology is the same across PortaPatch up to SyncroPatch, we are now having people who are trying to adapt this to a high throughput screening. So we had the opportunity to work with a company called Anatomic. And Anatomic is a producer of iPSC-derived sensory neurons, and their product is called the Real DRG. And what they do is similar to what people traditionally do with iPSC, generation of iPSC-derived cells. They're using a directed differentiation, and what they've been able to do is speed up this process and bring it to very large scale, so they can produce billions and billions of these iPSC-derived sensory neurons. And so this directed differentiation they can achieve very quickly within 1 week with a mix of defined media conditions, matrix proteins, small molecules, and recombinant proteins. At that stage, after 1 week of directed differentiation, they can freeze down those cells en masse and then ship them to customers. And so what we did working with Anatomic was to take the cells at that stage and then put them into culture using their the media and the coating for the tissue culture flasks that they sell, and then do further differentiation. So if you see this graph on the right here, the HIPSC stage is that's at 1 week of their directed differentiation when the cells are frozen down. We go and we differentiate or mature the cells further in our lab at Nanion. But just a little bit of background on these cells. At the molecular level, in terms of immunostaining, they show what you would expect for sensory neurons. So they have NAV 1.7. They also show marker for peripherin, which is a intermediate filament that's associated with the peripheral nervous system. A class 3 beta tubulin, which is also another hallmark of the sensory neuron phenotype. And importantly, TRPV1, because TRPV1 as the heat and capsaicin receptor is another hallmark of sensory neurons. Beyond the immunostaining, they've also done RNA-seq, and they show that 90% of the transcripts that you see in human DRG are found in their real DRG iPSC-derived cells, with the missing 10% being largely things such as transcripts that are found in immune cells or related to myelination and those types of things. But what we really wanted to do is ask the question, can we take these cells and achieve a high throughput patch clamp to sort of then interrogate whether these cells showed some of the hallmarks and characteristics of A, neuronal cells, and B, whether they're sensory neuron cells. So the goals of our experiment was to do a maturation study where we run full chip 384 well assays on cells after further maturation from 14, 21, or 28 days. We just wanted to see how well these things patch. It's not as simple as patching from an engineered cell line because in all cases we need a suspension of single cells for the automated patch clamp. So how well we can dissociate these cells is an open question. We wanted to just get a survey of the basic voltage-gated channels that are expected to be found in neurons, the sodium channels, the potassium channels. In nociceptor neurons, it's known that there are TTX-resistant channels, so they could be NAV 1.5, 1.8, or 1.9. We wanted to see if we can detect resistance, a population of sodium channels that are resistant to TTX. And we wanted to take a little bit of a survey of possible ligand-gated channels such as P2X or GABA, and then generally look at excitability. Does this change over the time in maturation? So the first figure here is showing an overview of the success rates. The green wells are the ones that pass the QC criteria. And my QC criteria here are membrane resistance of greater than 300 mega ohms and a capacitance value of greater than 2 picofarads. And if you look at most of the green wells, you'll see most of them have what looks like a nice fast inward sodium current. There are very few cells that show essentially no current. And the protocol that we're using here is a holding potential of -90 and just going through a series of steps. When you look at some of the individual wells, you can see that they show clear, fast, inactivating inward currents and more sustained outward currents that we presume are potassium. we're pretty certain, because when we can perfuse in cesium into the internal solution, those currents are decreased. What we see in terms of the cells that are expressing these channels is that the vast majority express both sodium and potassium channels, and that there's a little bit of a trend over time for a higher fraction of the cells. So you're going from, like, let's say 94% of the cells show sodium currents at day 14 up to 98% at day 28. But bottom line is they pretty much all show sodium and potassium currents. The next question that follows after this is we can do an exchange to remove the potassium currents. We exchange from a potassium-based internal to a cesium-based internal. And then on top of that, we add 500 nanomolar of TTX. That blocks, very efficiently blocks all the, quote, TTX sensitive channels, like 1.2, 1.7. And 1.7 is the predominant subtype in these cells. And if we see any current left over, we're pretty sure they have to be either 1.5, 1.8, or 1.9. And in fact, that's what we see. The blue, we're looking at the average current density over time. So at day 14, day 21, and day 28. In blue is what is before application of TTX, and the orange is after application of TTX. So in all cases, we do see, some TTX resistant currents. If you look at the percentage of cells that display at least some TTX resistant current, you again see a trend towards an increase over time in maturation. So about 80% of the cells show TTX, clear TTX resistant currents at day 14, and going up to about 94% on day 28. And the fraction of current on a per cell basis also has a trend towards an increase. About 9% of the current is remaining after application of TTX for cells that are at day 14, and about 13% of the sodium current is TTX resistant when you get to day 28. We can look at the V-half values for activation of the TTX resistant currents. And one thing I'm not showing you here is that a lot of the cells have very large sodium currents in before TTX application, and that gives us trouble with clamping. And so many of the cells are not clamped well, and so we would not be able to accurately measure a V-half of activation. After application of TTX, we actually have a much better time getting good voltage control, and so we can actually look at the V-half for activation of these TTX resistant currents, and we see, interestingly, that there's a little bit of a shift towards a more hyperpolarized V-half, and that's something we're going to follow up. We hope to follow up with in future experiments as we continue this collaboration with Anatomic. Switching to current clamp. So that's another easy thing to do, is just make this, after you go whole cell, switch into current clamp mode. And the first thing you can do is measure the resting membrane potential. And so down in this panel here on the right, we're comparing the results we get for resting membrane potential for cells at day 28 on the SyncroPatch and the patch liner. And we get a value in the range of -25. So it's a little bit, the average is a little bit more depolarized than what you might expect for a neuron. We're not exactly sure if that represents some sort of effects of the dissociation or not. But the nice thing is that both the SyncroPatch and the patch liner are showing complementary results. We're getting the same values. When we go into this mode, we are not injecting any current at all. And in some cases, we do see spontaneous action potential activity. And but it's at a very low rate. So at day 14, we have less than 1% of the cells display some spontaneous activity. And the maximum percentage of cells displaying any sort of spontaneous activity is about 3.5% at day 21, 28. And there's a clear coincidence here. If you look, I've just putting a blue marker in these graphs that show the spiking behavior. And if the membrane potential is getting down into that -40 to -50 millivolt range, those are the cells where we see spontaneous activity. All these other cells where the membrane potential is more depolarized, it's probably the case that all of the sodium channels are in the inactivated state, and just simply the cells are now quiescent. We can then, however, switch into a mode where we're doing a current injection to push the cells to a more normal resting membrane potential, in this case, minus 80 millivolts. And we do this with a low frequency voltage clamp routine that's built into the circuitry of the SyncroPatch. So we can keep the cells around minus 80 and then run a protocol such as the staircase protocol that I'm showing here, where we're stepwise doing current injections in 25 picoamp steps from 0 up to 180. And what we see is that as we step up to higher and higher current injections, we start to see many of the cells will start responding by firing an action potential. In some cases, just a single or a couple of action potentials, but in many cases, we're seeing whole trains of action potentials that eventually, as you get to a higher and higher injection, will eventually peter out. And we can quantify this in a really basic way just by asking what percentage of the cells actually show evoked action potential firing. And that's, I think, the interesting part here is that we do see a clear progression of excitability from going from day 14 to day 28, where we have 42% of the cells responding in this manner up to about 77%. And again, if we put a blue line in here at minus 50, it looks like the threshold for where the action potentials take off is right around minus 50 millivolts. That sort of ties in with the spontaneous activity that we were seeing in the previous slide. A quick survey of the landscape for ligand-gated channels. We basically just ran two agonists that would be activators for the P2X, which is be ATP, or for GABA, 100 micromolar GABA or 20 micromolar ATP. This is an example traces up here. So the first application is just an application of buffer, just to make sure that we have no responses to perfusion of fast perfusion onto the cells. So in both cases, there's really essentially no response to buffer application. And then we come in with either 20 micromolar ATP or 100 micromolar GABA. In the inset, I've blown this up, so we're just looking at the 300 milliseconds around that application step. And I should also mention that we can use the SyncroPatch to apply an agonist and give it an exposure time. So in this case, we're giving an exposure time of 1.5 seconds followed by that washout step. And so you can see a clear activation, some desensitization, but then a clear washout with GABA. In the case of the ATP application, there's a very rapid inward current that develops, but it also very rapidly inactivates in the continued presence of ATP. And if you look at the inset here, where I'm blowing up the time scale, you can see that over the course of 300 milliseconds, the response to ATP is completely inactivated, whereas with GABA, it's a much slower activation and the very minimal inactivation during that time course. We have just done a little bit of pharmacology here by showing that with a 15 micromolar GABA puff, we see clear and reproducible, and you could almost not see it because the black trace is completely overlying the blue trace for the vehicle control. But in the case of 10 micromolar bicuculin, you can see that we get complete inhibition of the GABA response. In terms of cells that show P2X or GABA, it's actually a very similar thing to what we saw with the sodium and the potassium currents. The vast majority of cells show responses to both agonists. And in this case, I don't really want to speculate on whether there's a trend one way or the other. I think it's just over time and maturation. There's not really a huge change. And I've also found out that there is there can be long-term desensitization and recovery from desensitization of the P2X responses to ATP. And some of my initial experiments, I think, were affected by that. So we would need to repeat some of these assays before I would speculate on anything like that. Okay, so this brings me to one of the final slides, success rates. So for cells that pass the basic QC criteria for seal and capacitance, we see a decrease over time. So we're getting about 56% at day 14. That decreases to 35%. That's a little bit disappointing, but not too surprising, because as these cells mature, they form more and more connections. Breaking apart those connections for the dissociation step becomes more and more difficult and a little bit tougher on the cells, I would imagine. But we're continuing to do some work on this, and I think we've already made some improvements to these numbers. And even with a 35% success rate, we're talking about having patched, more than 100 cells in half an hour. So it's still fairly robust in terms of throughput. Everything else here is showing that we have robust sodium and potassium channel expression. We do see an increase in the prevalence of TTX resistant currents. And I think the most interesting thing is we do see an increase in excitability over the course of this maturation. So in summary, we can actually patch clamp these cells on the SyncroPatch in relatively high throughput, success rate in the range of 35 to 55%. Reiterating that they all have sodium potassium currents, TTX resistant currents, GABA ATP responses. When I didn't go into too much detail, but the resting memory potential is more hyperpolarized by day 28. And what I did show you is the prevalence of action potential firing increases with maturation. I can also say that for all of these things that I just said, we found very similar results when we tested these cells on the patch liner. So for future work, one thing I didn't talk about was TRPV1. We still have not seen TRPV1 expression in these cells. We don't know exactly why, because the molecular work says it should be there. We speculate that we might be degrading it during the dissociation or losing it during dissociation. So we're actually preparing another batch of cells as we speak to try some different dissociation methods to see if we can figure out what's going on there. We also want to look at subtype selective NAV blockers to see if we can figure out anything more about the identity of the TTX resistant currents that are present in these cells. We've done a little bit of work with the GABA modulator, but we'd like to do a little bit more maybe with the P2X channels. We also have plans for looking at HCN channels, which by molecular results should be there, and piezo channels, which is also something that is should theoretically be there based on the RNA-seq data. And we've also, in parallel to this, we've been developing methods for using very rapid perfusion of solution onto the cells to activate piezo channels, at least in heterologous systems. So the hope would be that we can actually apply those techniques to these iPSC-derived neurons. And with that, I would say thank you to everybody for your attention. And I also want to thank everybody at Nanion, Rita, Rodolfo, Irene. Irene did all of the work on the patch liner. Aaron did some of the pioneering work on dissociation. Rita took care of the cell line, all the cell work and the culturing. And Vince and Patrick, the CSO and CEO over at Anatomic, who are great to work with. And any questions, if we have time, I'll be happy to answer.
Well, thank you so much, Tim. It was really fantastic. I've been at a Nanion conference about a month ago, and I learned something totally new today.
Oh, good.
Yeah, so it's very, very exciting. So, I have two comments to make. One is that we are also using these real DRG neuron with success, not with automated patch, but with manual patch clamp. And so, so they're very, very useful for research for the reasons that you have listed before, right? So they're very fast. So within a week from the moment you thaw them, then you can actually get mature neurons. So it's very useful, and they are transfectable too, which is another major barrier for primary neurons, primary cultured neurons.
Right, that's true. Yeah, and the one thing I forgot to mention is, part of the impetus behind the iPSC approach is that they're human, right? Sure. A lot of the work that people do are on rodent DRGs, and that introduces some uncertainties when you're doing therapeutics development.
Absolutely, and especially in the field of pain, people have experience a lack of translationability from preclinical rodent models to humans. So having a human platform early in your screening is very useful actually to de-risk compounds, especially if you do drug development or drug discovery. So I think this is really good. Yeah. So another thing that caught my attention, it was a very small thing that you mentioned at the very beginning, talking about the ability to assay GPCR, the protein coupled receptor with your equipment. What exactly, can you elaborate a little bit on that? There is an expert here on protein coupled receptor, Dr. Allen. So I'm just very interested. So what do you do?
So the answer is yes, but you asked me if I could elaborate a little bit, and I can only elaborate a little bit. So I'm not involved in the development of this is a new platform that we're developing, and it's going to be released. I heard it's going to be released soon. It's called the Atlas, and it's spelled with a Z, Atlas Z. It employs what they call spectral impedance, which is a technology that I have yet to completely wrap my head around. But they are an impedance signals. So the cells are on a surface where there's a layer of cells above a surface that has an electrode in it. And you can make measurements of the impedance, and that's where I'm going to lose all of my credibility if I try to say anything more than that.
That's okay, but so you would be able to measure any functional activities, functional signaling downstream of
that's my understanding.
Wow.
But I don't know exactly how that works. And I don't know how exactly it relates, because when I think about GPCR signaling, I think about, a lot of different aspects of it. There's the G beta gamma and there's the G alpha.
There's also the beta arrestin pathway. So
and I don't know if you're getting some sort of just rough general signal, or if that's coming from, one of the parts of the heterotrimer or another. Those are the parts where, boy, my brain really kind of shuts down, and I need to learn more about this. But I'm hoping that when this product is released, that they're going to have some pretty nice app notes to kind of highlight some of these different features.
Well, this is great. Yeah, something new. I didn't recall it even mentioned at the Nanion retreat that we had. So maybe they did. I just didn't.
Yeah, maybe we should keep this as like on the down low, because maybe I'm not supposed to be talking about it too much. I don't know. But okay, no, I think it is something that was mentioned. It just says,
it was mentioned. Okay, that's good.
This is something that's coming, and it's supposed to be soon. But I think it's one of those things that they're not going to do a release until it's like fully ready. Okay, so that's why I try to put it down there at the bottom. And
sure. I'm just curious. I'm just gonna shut up because I think maybe Dr. Allen or John will say something.
No, I was just going to say, I'm excited. I'll follow up, Tim, and keep my eyes open. That I'm the one that studies GPCRs, and it is all about neuromodulation and neurons, of course. So, and it's most often the G protein modulation of the channels and their kinetics and things. But there are impedance-based assays for GPCRs that, interestingly, what they pick up is they look at basically cytoskeletal reorganizations and translocation events that bring the bulk protein into sort of membrane aspects, and this will actually change the impedance of the membrane generally. And many GPCRs, through their second messenger, signaling cascades or recruitment of certain proteins, the impedance will change on the membrane. And there are different impedance-based assays that are basically phenotypic-based only on impedance. And then people use different sort of inhibitors of signaling molecules and things, and they sort out with impedance. But maybe it's something like that. And I know we were all speculating, but I can't wait to find out more, Tim. Thank you.
Okay. Yeah. So stay tuned for more on that later. Yeah.
Sounds pretty cool. Thanks.
Great. So any other question for Tim?
I have a question. I probably asked you this, Tim, some time ago, but the time passes, so maybe there are some new developments in this direction. So, has anyone tried to use SyncroPatch to measure channel activity in proteoliposomes? I can imagine that the liposomes will break, of course, upon touching the glass, but maybe there is a patch formed.
Yeah. So far, I'm not aware of anybody doing reconstituted proteoliposomes on the SyncroPatch.
So
pretty sure that's a no.
Tried, or they tried and failed.
I'm pretty sure that on the SyncroPatch, I'm aware of nobody that's even tried it.
Right. Okay. Yeah, because we have a user who actually plans to do just that.
Oh, really?
Okay. Oh, yes. But she said, okay, not right now, maybe after a couple of years or something like that. Mm-hmm. But I'm wondering whether it's possible in principle, because I can imagine that something like excised patch configuration might be possible. And she is interested in channels that generate really huge currents, IP3 receptors. Mm-hmm. So I'm hoping that maybe there is something in this.
I hope so too.
Whether anybody tried that. Yeah.
No, the things that people are trying right now are patching of lysosomes, patching of mitoplasts. Those are the two main things that I'm aware of. And so people are working on that. But in terms of reconstituted, I'm pretty sure that it's not been tried, at least not to my knowledge.
Yeah, because, well, definitely, they will definitely burst, because I remember like when you just touch it with a pipette, it blows up. Yeah. But what remains, the question is what remains there?
Right.
And if there is a right patch remains, so that would be interesting. Okay, thank you. Thank you. Fantastic. Yeah, by the way, another question, very brief. Mm-hmm. Do I understand correctly that these real DRG neurons, they do not need any glial layer?
That's correct.
They just grow on glass.
Fantastic. So there's a special coating
of course,
that they provide. Yeah.
But no glial layer.
No glial layer, no.
How expensive are they?
They're fairly expensive, and I can't remember the exact cost. Fernanda's nodding. She could tell you.
I think they're about, what, 1,200 per vial, something like that. But then you can get a full plate with them. So, but I think they're really worthwhile, the cost. And the reason is because, at least for our purposes, we were trying to reintroduce or introduce either shRNAs or, cDNA plasmid for gain of function, loss of function, and we were looking for a way of transfecting them, and they can be transfected we use a magnetic beads base method, and it works very well. So, I mean, they cost a lot, but hey, in a week you get the neurons.
And anyway, getting neurons is costly. So maybe I should learn something from you about this.
Okay, good. So
$650 for a while.
Someone from Mm-hmm.
No, it's a reasonable price, I would say.
Or $1 million. I don't know why. Yeah, maybe yes. Mm-hmm.
Maybe we should talk with you later, someday later, not right now.
Sure.
About this, because this looks like it's a good option to have.
Absolutely.
Neurons.
Yeah, I'm hoping that Anatomic will also consider doing cortical neurons, right, rather than, or other CNS neurons. And I know maybe they're working on it, but I don't know. Those are a little bit more difficult. We have tried with another company to get these cortical neurons. They are not as good as the real DRG. So I don't know if it's a matter of different neuronal subtypes or if it's a matter of different proprietary, soup or recipes that the company provided from different companies. I don't know that, but I'll keep you posted if you want.
I'm very interested. I'm very interested in that because, we obviously need some workable neurons for our studies. Yeah.
So, any more questions from the audience?
May have one.
Of course.
So, Tim, first of all, congratulations for the talk. It was amazing. And so, a curiosity.
So can you measure in cell attach configuration with the SyncroPatch? And if yes, is there a way, or better, are you planning to develop a system to control the stretch of the membrane for studying K2P channels? You know, there is a community of people that would love to have this device to screen drugs, compound, or whatever on their channel.
Yeah, so that's a great question. I would say the answer is it's difficult to do. And it depends in some part on the type of cell that you're using. So a lot of engineered cell lines are HEK based. And for whatever reason, HEK cells compared to, let's say, CHO cells have a high probability of entering the whole cell configuration spontaneously. So when we actually capture the cells on the chip, we are applying a mild suction. And sometimes that mild suction is enough by itself to cause the cells go into whole cell configuration. So that's problem one. And then the other issue is with the amplifiers on the SyncroPatch have low and mid gain settings. We don't have high gain settings for the SyncroPatch amplifiers. It's my understanding it's cost and space prohibitive to do so. Yeah, so the getting down to single channel in the cell attached, I think would probably not be feasible unless it's a very, very large conductance channel.
Yeah, but maybe also a macro current from a cell attach is enough.
That I think that could be a possibility, and I think, but what you want to do is find the cells like CHO cells are less prone to going into whole cell configuration, so that might be an option. But there may be other cells out there that are even better choices than that. But I think that would be the first fundamental thing that you would want to do.
Or maybe just playing with the whole sides. So maybe reducing the size of the core where the cell sits is the solution.
Yeah, it could be. In fact, we are the people who are doing the lysosome patch, we're working with them producing custom chips that have ultra small holes to accommodate those small organelles.
Thank you, Tim.
Sure.
And Fernanda, may I ask you a small curiosity? In your hands, what's the efficiency of the transfection of the DRG neurons?
Oh,
roughly. With the magnetic beads system that you were mentioning before.
I don't want to say numbers that are incorrect. There are sufficient to do ample number of experiments, but I actually don't. I don't want to say any number. Definitely it's not like a lentivirus, right? I think those viral based, you can just infect everything. Definitely not that.
But enough to do something, let's say.
Yeah, I will have to get back to you with the real numbers, because I don't want to say something that is totally wrong. Because I don't know. If there is anybody from the Laetza's lab that has these numbers, maybe right now, I don't think, the primary scientist using this these cells is not here at the moment.
Someone in the chat has wrote 20-30% for a good transfection.
Okay, thank you, Nolan Dvorak. Maybe you can just say a few things, Nolan. And
I would say the transfection, it varies a lot. I mean, there are sometimes you do it, you get less than 5, and there's sometimes where it goes really well and you get 20 to 30%, but there's a lot of variability, and I'm really not sure what's at the bottom of the variability of that transfection efficiency. But yeah, I mean, it does work pretty well once you kind of get the protocol optimized for it.
Thank you. Thank you very much.
Yeah, absolutely. And I see in the chat people that are sending direct messages about this method. Absolutely, I can provide. I'm trying to reply here, and I don't know why I can't reply to this person. Anyway, I'll provide information on this magnetic beads for sure.
Great, that would be very good.
Thank you. Sorry, I don't know why I'm trying to reply to you. Oh, okay, yes, okay, got it.
It's okay. It's okay. If you can speak, you don't need to use chat.
Yeah, okay, absolutely. So. Anyway, I think it was really great. By the way, we are going, the Neurotherapeutics Workgroup is part of the Mental Health Research Consortium. We're going to host our annual conference in September, and Elements will be there. Nanion has indicated an interest, but has not finalized yet in sponsoring. So usually we have, so the Mental Health Research Consortium has now over 200, actually it's more, almost, I think 250 or so PIs, principal investigators across the Texas Medical Center with a wide variety of interest in mental health research, starting from the drug discovery, basic science, to the clinical side. So we should have this final agenda up. It should have been up now. I can check, but so that at least Nanion can consider whether, what type of involvement they want. I know speaking with Rodolfo at the meeting in Boston, they were committing by maybe the lowest level to begin with, but potentially upgrading. So it's a great exposure because it's in the middle of the medical center. In addition, having already a SyncroPatch and 2 PortaPatch at least on site, it will be really a great tool for advertising. Because at the end of the day, it's the cost of the plates that makes Nanion rich.
Rich.
Yeah. So, and again, thank you so much, everyone. And thank
you for organizing it. That was wonderful. And we should repeat it from time to time.
Absolutely. Absolutely. Thank you, everyone.
Okay.
Bye-bye.
Thank you. Bye-bye. Thank you.
Bye-bye.
Bye.